Hello, it's Mr. Singleton, one of the Associate Directors for Outward Academy Trust. This is, again, a lesson for Biology Paper 1, and this is one of the required practicals. It's required practical number 4, and it's food tests. So this is the specification. So you have to be able to test for carbohydrates, and within carbohydrates, it's sugars and starch, lipids and proteins. So this is just a bit from the specification, and it just helps you to remember what you need to know. So I just want you to have a think about when you've maybe eaten a packet of crisps or anything like that, looked on the back of the packet of crisps and there's something called a nutritional table. Can you just have a little think, I'll just give you a minute, about all the nutrients that you might find on the back of that packet of crisps, about what nutrients are in there. I'll give you 1 minute, off you go. This packet or a nutritional table from the back of a packet of something. So we've got energy in there, protein, carbohydrates, fats, fiber, sodium. They're all types of nutrients. Now, sodium is an interesting one because it'd come under a mineral. So you've got vitamins and minerals. This food obviously didn't have any vitamins in at all. So title for today's lesson is Food Test Required Practical number 4. So you can write that in your book with the title and the date and underline it, or onto a revision card if you're using revision cards. These are the challenge and aspire. So the challenge is to identify biological molecules, so those nutrients using these reagents, and then the aspire is being able to write the method and a risk assessment. So when we do an experiment, what we do beforehand is we'll write a risk assessment to cover all the risks that could happen in the experiment and how we can control that and stop them being so much of an issue, so we stop any accidents happening. So as teachers, we write those risk assessments and we have them. If you're ever asked about a method, it's usually a 6-mark question in the exam. Try and think about MAV, okay? And that stands for method, apparatus, a risk assessment, repeating and labeling those variables. I'll talk about variables in the next couple of lessons. But you may have come across independent variable, control variable, and dependent variables. So if you are asked for a 6-mark question, write the method, list the apparatus, a risk assessment, repeat, make sure you talk about repeating them, and then list your variables. You'll do very well on those exams. Straight to the point with this one. So if I want to test for sugar, I would use a chemical. If you look at this table, I'd use a chemical called Benedict's, and all I have to do is I make sure that Benedict's is in I make sure that my food is in a solution. I add my Benedict's, and then I put it into something called a water bath. And in the next slide, I'll show you that water bath. And what you will get is it'll go from blue and then to orange and then to brick red. So if we're talking about risk assessment, that water bath can get really hot. So I can turn it up to like 95 degrees Celsius and I can leave it there. So the risk is the water bath gets very hot. How to control it is just don't leave it in there for too long. All right, and then you'll be okay. If I'm thinking about this, I always think about Lucozade Sport. Lucozade Sport says glucose on it because it's got lots of glucose in. The sticker on the outside of Lucozade Sport is blue and the contents is orange. So I just think about this Benedict's working its way in, going from a blue colour and then to an orange colour, brick red if there's lots more in there. Here's a practical for. So in order to do this, I'm just going to use 4 different foods, sorry. So in this one here, in this test tube, I'm going to put some corn flour. Right, I've just labelled it CF pretty badly. I'm just going to add my corn flour to this one. Right, I'm going to make sure I use a different spatula every time. So my corn flour's gone in there, rather badly. I'm going to put some powdered milk. Okay, that's this stuff here, this powdered milk, into this test tube. I'm going to put some glucose solution into this one labelled G. And using a separate spatula, I'm going to put some protein powder into this one here. Alright, so what we're going to do is we are going to test for sugars. And for this, what we need to do is to make these powders into a solution. So all I'm going to do is add some normal distilled water into each one of these test tubes. So it's just to get it into a solution, because the solution that we're going to need needs this into a solution. And the solution I'm going to use is this blue solution called Benedict's solution. Okay, so I'm going to add a couple of drops of Benedict's, which is blue at this point, to each of my test tubes. So there's some into my corn flour, some into my milk, some into my glucose solution, and some into my protein shake, which is brown, which is going to be a bit of an issue maybe. Just need to add a couple more into there. Now we're going to use this piece of equipment called a hot water bath. This needs heating up. Okay, so there are 2 ways to do this, 2 ways that AQA would like you to know about. This is my hot water bath, this is my machine, which keeps things at a constant temperature. If you look at the required practical about enzymes, we used this piece of equipment before. This dial here heats up the water which is inside to the required temperature. So it's like an oven. You see that switch is on now here. Okay, this light is on here, saying that it's going to heat up. But once it gets to the required temperature, in which case here it's 75, then that switch will turn off. I've got a thermometer in there just to make sure it's checking that it's working, and I can put my solutions into this hot water bath. All right, and they will heat up. If for some reason you don't have a hot water bath, you can make a hot water bath, and AQA like you to know about this. So to make a hot water bath, it's very, very easy. You get a big measuring jug, and you are going to add your hot water straight into there. Okay, so you add the water that you want. and then you can just put in your test tubes, okay, and lay them in there, making sure that they don't spill in. So now that is just laid in there, and that's a hot water bath. It's just to get the contents that's inside this test tube to the temperature without just adding lots and lots of hot water. So this is a hot water bath, but also you can use this piece of equipment as a hot water bath too. So Benedict's to test for that sugar, and what should happen is you should see a colour change once they are up to temperature. I'll just give them a quick check. That one still looks blue, that was the milk. This is the glucose, you can see the glucose now. Here is just turning slightly green at the bottom there. If I just give that a shake just to make sure that sugar's dissolved, we'll put that back in, nothing else happens to it. And the corn flour there is still a very light blue. Give that one a good shake too. So we just need to leave them in there and note any colour change. So there are different ways to test for your sugars. And remember, it's Benedict's solution, and it was blue at the beginning, and what we are looking for is any colour change at all from blue. So this is the milk, this is still blue. If we have a look at the glucose syrup, all right, it is slightly changing to a light green. We'll leave it in there a little bit longer, see if it changes a bit more. So if you just look at these now, they've been in there for a while. Okay, this one here, which was corn flour, has stayed a blue colour. My milk has also stayed a blue colour. But my glucose, okay, because it's obviously got sugar in it, has gone this orange colour. Okay, and that's one of your positive tests to show that there's glucose there. My protein has stayed the blue colour. So that is a test for glucose. Use this blue Benedict solution, and then you add it, but you need to use a hot water bath, and then you should get the colour change to yellow. In order to test for starch, which is another carbohydrate, then you just use iodine. This is quite a simple one. You take the iodine, place it onto the food, and then the solution, which is originally orange or brown, goes blue or black, as you can see from the potatoes on the next one. The risk in this one is that the chemicals can get into the eye. So in order to limit this, you'd obviously wear safety goggles and use iodine solution and not pure iodine. So here's the test. It's quite a straightforward one. So this is part of the food test required practical, and this is how you would test for starch. So again, I'm using one of these spotting tiles, this white thing here, spotting tile, and I've put some different foods in. So this white one here is corn flour, this is protein powder, this one is some powdered milk, I've got some bread here, I've got my son's favourite crisp spicy Doritos here, and then I've got smash, which is like instant mashed potato, and some glucose sugar, pretty much. So the first thing I'm just going to do is I'm going to test these for starch. Now, this is quite a common one because we used it in the magnification one when we were looking at cells using a microscope, and we also used this in the enzyme experiment. So we're going to use this thing, which is iodine. Now notice it is brown at the moment. If you see that liquid there, it's a brown. If starch is present, it should turn black. So this is a very simple experiment. Although once you start to learn all the food testing reagents, people start to forget. So all I have to do is place this onto my food, and if it turns black, see how that's turning black, then it has starch in it. I'm interested in this one because this is my protein powder. I wonder if it has got starch in it. It doesn't look like it. Milk stays a nice brown. The bread turns black because it's got lots of starch in. The Doritos will turn black because it's got lots of starch in. Potatoes turns black, but the glucose will not turn black. All right, so you can see that these, if I just give it a mix, this one's turned a very, very black colour. Okay, but if I go over to the glucose, just give it a swirl around, that has stayed brown. So if it turns black, it's got starch present in it. If it stays this browny colour, then it has not got starch present. That's the starch experiment. Iodine turns black if starch is present. So I want you to have a look at the Google quiz now, see if you can answer those questions on those two tests. Off you go, I'll give you 4 minutes. Okay, you got 1 more minute. The next food or nutrient that you need to be able to test for is protein. And protein is tested for using Biuret reagent. And this is again quite an easy one. Just add a couple of drops of Biuret and it goes from blue and into purple. The risks for these are if it gets into your eyes, you need to wash it very quickly. And sometimes the colour is really hard to see. So sometimes you can use white paper underneath the solution to make it more obvious. So here's a video of it to help you out. This is the food test practical, and this is the test for protein. So what I've got is I've got some albumin, some protein shake, some corn flour, some glucose and some milk here, powdered milk. And all I've done here is I put them into a solution into these test tubes. So I've just added some water into each of these test tubes and then added the albumin, the protein shake, the corn flour, the glucose and the milk. And to test for protein, we're going to use this, which is Biuret reagent. Okay, so notice it is now blue. And what we're going to do is we're going to add a couple of drops of Biuret to each one. so it goes a blue colour so I can see it. A little bit more in this one. And into this one. And then I need to give each one a good shake. Okay, so I'm just going to shake these really well. Put a bung on top of it, just give it a quick shake, and then leave it to settle. Here's my protein shake one. Not convinced that one's going to work because of the colour of it. And the milk for the last one. Okay, and all I need to do now is leave them to see if there's any colour change. So we've left these for a while now. We'll just go through these results. So if you can see this one here, which is the glucose syrup, that has not changed at all. Okay, it's still that light blue colour, so that has not got any protein in, surprise, surprise. If I match that against this albumin here, and you see the colour change there, this one's got a purple colour, which means it's got protein in, which is not a surprise because it is a protein. So that is your colour change. It goes from this light blue here to this purple colour here, and that shows you that the food has protein. Now if you look just very quickly, this milk has a little bit of protein in it. It has got a purple colour, so it does have protein in, but I'd expect that from milk as well. And the last food test you need to know is the test for lipids. And there are actually two ways to do this. I've put this one on, which is sedan-free. And what you do is you add a couple of drops of sedan-free to the solution. And what you get is this red layer that floats on top. Now it is flammable, so you have to keep it away from any Bunsen burners. The other one that you can do is, I've shown you on the video next, is you can just add ethanol to the solution, shake it, and if it goes cloudy, it's got lipids in. So there is two for this one. You can use either one for AQA. That's fine for your exams, but I've shown you both tests here. This is the required practical part of the food test one. It is for lipids, also known as fats. So what I've got in this test tube here, I've got some glucose in, which has just come from here, and then in this one, I've taken some butter. Okay, what I'm going to do first is I'm going to make them into a solution, or at least try and get some of this fat dissolved. I'm going to add some water to each one. So I'm going to do it about a third full in this test tube, and into this test tube. And then what I'm going to do is I'm going to give them a good shake, because I want the glucose to dissolve, otherwise you'll end up getting a cloudy solution either way. Now there's two ways to do this one. I'm going to show you the ethanol one first. This is the one which is probably the easiest. Okay, so I can just add ethanol to each one of these test tubes. And into this buttered solution here. Okay, and if the solution turns cloudy, then fat or lipids are present. So give this another quick shake. You can see that this one is not very cloudy at all. I'm going to give this one a shake. Okay, it's very cloudy. Now to double test that, if you want to improve this a little bit, I've got two test tubes with just some more distilled water in. If you take this out here, and then you add it, and if I take this one out, I add it, it helps you to see the difference a little bit more. Okay, so this one here was the butter one, which has definitely gone cloudy, and this is the glucose one, which hasn't got cloudy. So the positive test for a lipid, add ethanol to your food, shake it, and if it goes cloudy, then lipids are present. The other test that you can do for lipids or fats is that you can add this substance called Sudan free. Okay, and all you need to do is put your solutions you are testing. So I'm going to put in some glucose into a test tube and add some water. About a third full will be enough. And then I'm going to add some butter into this one here. Always hard to get butter into the actual test tube. Sticks to the side. There we go. So my butter is in my test tube with some water in again. And then what you will do is you add your Sudan free to the solution. So here's my Sudan free in this pipette. Add it to that solution, and then add it to this solution. And this is the difference between a fat or something that's got a lipid and something hasn't. This one does not. It's just turned the whole solution a red colour, whereas this one here has got a red band at the top. So a red band floating on the top of the solution means that it has a lipid present, and that is how you test for lipids. So they're the 4 tests. I just want you to go back to your Google quiz, make sure you understand them. So go through them last couple of questions on those food tests. I'll give you 3 minutes, off you go. You've got another 30 seconds. So that is both the challenge. You should be able to identify the reagents used for each test and then be able to write the method for each one and assessment. Again, if you're doing revision cards, this is B1. So So it's biology paper 1 that'll be sat probably in May. So write that at the top of your revision card. So starch test, all you need to do is add iodine and it will go black. The protein test, you need to add Bio-Reactor reagent and it goes from blue to purple. The sugar test or glucose test, which is a little bit harder, you add Benedict's, put it into a hot water bath, and then it should go orange and then brick red. The lipid test, you can use either of these, add ethanol and it goes cloudy, or you can add Sudan-free and a red band appears at the top of the solution. Please, if you are doing these in the exams, remember that starch and glucose or sugar are both classed as carbohydrates, and then protein is by itself and lipids are by themselves. And don't forget to mention the colour that they originally were. So remember, starch test, add iodine, which is brown, and it goes black. I'll give you 2 minutes to write those down. Again, if you need to, just pause the video and then you can write them all down. But it's a good idea to have those notes for you. There are some exam questions for you, so if you print them off and complete them and put them into your book, or try and do them online and then pass them into your teacher for marking, just to help you with any of those questions that you might struggle with. Well done.